I think we should start because other people will be joining in 4 to 5 minutes. So we can start. Start. Uh, is my screen visible to everyone?
Yes. Yes, sir.
And also the marking, right?
Yes, sir. Yes, it is visible.
We are starting. And is there any option that some other person can accept members who are joining? I guess there are 31 already. So correct. So this is a basic introductory session, so that we understand what we will be learning in coming sessions. Like first, let me clear what we are going to do in next 6 months. These will be like short sessions of 30 to 45 minutes every day, and I will be sharing examples and other things. So for 6 months, and we will also do some sort of case studies, and whatever research is published, we will take example from there, so that you understand how to understand the dynamics, molecular docking and dynamics, and how they are presenting their data about docking and dynamics in their papers. So uh, and we will try to replicate all those research papers which we will be analyzing, and at the last we will be providing certification after the hands on. So my first question is, we all are from the background biology background, so we understand what is a molecule, like protein, DNA, RNA. Can you example other molecules as well? Not audible.
Carbohydrates in there. Lipids.
Is there any problem? Okay, lipids, carbohydrates, and signaling molecules like ATP, ADP, and other molecules. So in the biological system, these molecules play their role in processes or some other interactions. So what we do is we need to understand those interactions, And one thing I want to make you understand that there are large number of proteins, like if you see protein data bank or some other in vitro system where list of protein is given, we see there are millions of proteins. And some and DNA has around 3 to the 3 billion base pairs. And there similarly there are more than 3 3 millions of RNA sequences. And lipids are also very high in number, carbohydrate also, and other signaling molecules are also there. So to understand how these molecules are interacting in a in vitro system or in a cell to produce some effect or phenotype, or even some how they are driving some reaction biological process, we need to understand how these molecules are interacting. So what we are going to do is that we will understand how these interactions are being performed or deciphered in the research field. And for that, we can't always use in vitro system as they are very costly. So we are going to go through a basic part of in silico system, in silico studies, that is called docking and dynamics. So basically here we have few understandings that why are we going to use molecular docking and dynamics. First is that performing in vitro experiments or experiments on cells or cell cultures is very costly. Going to be costly if we have to understand thousands of molecules and understand interactions of thousands of molecules at once. Like I have already told you, the number of there are millions of proteins and billions of uh, nucleotide base pairs for our genome. So all those things will take too much experimental expense. So for to overcome that, we go for in silico first, then we carry out that in vitro system. So this in silico proves to be cost effective. And basic motto of the molecular dynamics study is to know how two molecules are interacting, biomolecules are interacting. Based on that interaction, we are going to predict some new inhibitor or drug molecule which is targeting, or even some new molecule which is enhancing or altering the biological process. So main motto of the docking is to understand that interaction and save the cost of in vitro experiment. Next is we need to understand how existing interactions are working. Like if we talk about a cell on which there is some receptor and some signaling molecules comes to it in its contact and triggers some response. So how it is going to interact with that? So to understand that interaction and how it is going to trigger further response, we are doing molecular docking. Okay, so the next is uh, what actually we study in how we perform molecular docking and dynamics, and how both of these things are different. So one basic question is that if there is a cell, and we assume that there is some receptor molecule or a molecule inside the cell, and if another molecules like some signaling molecule or a drug comes in its contact, it takes it inside, or if some molecules enters through some channel or some receptor protein or some carrier protein, and it goes inside the cell and in the cytoplasm. So what it is going to do, it is going to interact. So how that interaction is deciphered, we in in vitro system like we we first perform that perform that experiment. After that experiment, we extract that complex. First we perform experiment, then we extract complex and apply some in vitro methods or complex methods like NMR or X-ray diffraction or some these are very expensive method, and we use those methods to identify how these things are interacting. And based on collecting all these links, we we understand that how a biological process is working. Like if we talk about a signaling pathway, first we need to understand that a receptor and drug is a drug or a signaling molecule is interacting, and later this signaling molecule is going inside, and there is interacting another signaling molecule which is interacting with another receptor, and that receptor is binding to some intranuclear region, and then triggering some other response. So these are linked through various experiments and validated. Then we get too many publications, and those publications let us understand how things are working actually. So these if we understand these experiments quite well, then these are very expensive experiments. Like we have to have good quality of uh, lab reagents, and using those lab reagents uh, might be very costly. So to overcome that, we what we do is we simulate those molecules. Means simulate those molecules means if we understand about structure of a molecule, there are atoms. Okay, so to understand that how a structure is there, and how we understand the interaction, first we need to understand what is the structure of a biomolecule. Like if we take any biomolecule like protein, DNA, or lipid, there are atoms bond together which form basic building block or unit. For protein it is amino acid, for DNA it is going to be nucleic acid. Fine. And those units connect together and form that molecule. Like for 2D structure there are some signature hydrogen bonding, and for 3D structure there are some other type of hydrogen bonding, and then there is quaternary structure. So protein, DNA, or any other molecule basically follow this pattern to show their activity. So what we are is how can I clear this? So since fine. Any way to clear the ink? Make it. So as we understand that amino acid nucleic acid are going to form the three-dimensional structure of DNA or some protein. Then there are atoms, and those atoms are constituting amino acid, or inside DNA they are constituting nucleic acid, and together they are forming some chain to give the three-dimensional structure after hydrogen bond interaction. This is our in vitro result, which we are already understanding in our basic studies like in BSc or even in 10th or 12th class. Next is, this same thing is going to be incorporated in silico. In silico, we have atoms like carbon, hydrogen, nitrogen, or whatever element is going to be the part of that particular structure. And this is one information. And carbon has some charges or electrons near it, and hydrogen, similarly hydrogen or oxygen. So like in chemistry, we have studied how oxygen interacts with hydrogen or carbon in a amino acid or nucleic acid. So that information is translated into in silico system using these symbols, and these symbols are further taken to some other program where this docking process actually happens. So so this makes our life easy in terms of in vitro experiment, and reducing that cost by simulating molecules, what to say, by simulating those molecules. So by simulation of those molecules, we can further study the in vitro interactions through some software which are already built. And we also understand that in chemistry we already have uh, atomic properties, like one atom, if one atom is binding to another atom, then what is the distance, and how those are interacting, like what is the charge between them. And if if we take example of hydrogen and nitrogen, there nitrogen is electronegative, so it will pull charge of hydrogen. So all that information is used in silico. We will be dealing with this by visualizing and actually hands-on through software and packages. This is I am just giving you overview, just what you will be learning through in silico or docking and dynamics program. Next thing is, what is the difference between docking and dynamics. I am just giving you brief. While performing the hands-on or other things, you will clearly understand all these things yourself. Basically, docking and dynamics are not much different, because we understand interactions there. Docking is simply, we take one molecule that is called receptor, and we find interaction with another molecule, that could be any other molecule, which could be a ligand, protein. That ligand could be a protein, DNA, RNA, or a small molecule, or drug. And similarly receptor could be a protein, DNA, RNA, ligand, or any drug. So what we do is that how that receptor is going to interact with that ligand part, which is another molecule. So how that is going to form hydrogen bond, or uh, how elements or residues of that particular receptor are going to interact with another atoms of the ligand. So that we study in docking. And in dynamics, what we do is, we check how long these interactions are going to survive. Like if we take a timeline, like if this is some cell, a receptor, a ligand is coming, and there is a receptor. By docking, we understand that this particular structure is going to interact with that, and it is going to perform some interaction using van der Waals forces, hydrogen bonds, or some salt bridges, or through hydrogen bond linking, or any type of interaction we study in chemistry. So if that is going to interact, we are understanding, we are getting this understanding from docking. And how long is actually this is going to last is from dynamics. So at suppose time 0, when these two are interacting, next time sometime t2 t1, this might be like trying to find some binding site. So that molecule will be moving, maybe it might be changing its interactions, like find forming other types of bonds, or breaking some existing bonds. So there are a lot of things happening which we understand through dynamics process. So over the time how these interactions are changing, we study all those things in dynamics. So basic basic motto of this molecular docking and dynamics will be handled in through this session. And where we will be starting with docking, and later after docking we will be going for molecular dynamics. And all these things will be and all these things will be relevant to your research. Means that is your time given in these sessions will not is not going to be waste, because I will be handling all the case studies, multiple case studies, not all. And by hands on. Am I audible, or I'm just continuing speaking, because there is
Yes,
a
sir,
lot
you
of
are audible.
Yes, yes, yes. Yes, you are audible.
okay. So after the sessions, what you will be getting is I will be like starting with a very basic, like how to understand the structures, and how to understand the structure and how to study their interactions. Later on, those interactions will be understood through dynamic simulation, and I will be mostly using some GROMAX or some software. I will give you the list of all these things uh, by step, and you will be learning everything from very scratch. So all the resources will be provided, and you will be given sufficient time. Like if I am going to perform some software visualization, I will help you understand install that software. After that you will be given some time to perform that visualization yourself, and feel how it is going to work. If there is any problem at any step, I will be taking the queries and doubt session, so that or instantly I will try to solve your all the problems. Session will be late, but my motto is to enable you to solve your problems yourself. Means you will not have to depend on others for your docking and dynamics part. These sessions are going to be very short. Like in the next session I will be dealing with installation of visualization softwares only. Many of you will be knowing many visualization softwares like PyMol or Chimerax or VMD. All so I will be dealing with all these softwares, and help you understand how you can do the basic things. And after those basic things, we will be moving to docking part. And after docking part, we will be visualizing the interactions after docking. So it will be like you will be visualizing those interactions using these software. And like this you will be having the list of all the maximum resources which you could use for your research, or your publication, or your for project proposal, if you people are in MSc or going to propose uh, uh, for training purpose or some other things. So you will not have to be dependent on other training or training resources for talking. It is my assurance. Basically, these hands-on training will be helpful to you to perform all those things yourself. And next will be we will be dealing with some case studies, like we will be taking some research papers where interactions are reported, or we will be taking some in vitro examples where interactions are present, and how we can actually visualize those interactions in our in silico system to to propose some new hypothesis, like how one molecule is interacting with another molecule. So all those things will be done. And I will be mostly available through telegram for your doubt solving session or throughout the week. But there will be only two sessions where I will be breaking down all docking and dynamics in very small parts, so that you can practice for a whole week, because most of us are already taking some college or lectures or doing some research work. So these small sessions will be very helpful. And in the last, if I guess if certificate is important for you, we will be providing those certificates from Biomix group. So it is going to, maybe it will be helpful for you to have a good some sort of CV, helpful in your CV. As already discussed, I will be starting with the molecular visualization thing in your next session. So we will be starting with actually biomolecular structures work, and how you can read all those structures, read, view, or alter structures. Like you might have heard that there is some mutation in protein, there is some mutation in DNA, or some other structure. How you will be visualizing all those mutations or changes or alterations, and how you can actually perform those alterations yourself, and how you can so these basic understanding will help you in that direction. Next is since we are going to understand all these structure for visualization, we will be having some set of tools, because different tools have different capabilities and different easy things. Like if we talk about VMD, it is very helpful in uh, visualizing molecular dynamics, how molecular dynamics, or how two molecules are interacting over the time. And similarly chimera X or pymol are very good for understanding how docking results are shown, like one molecule is forming what kind of bonds in after docking. So that all those resources or visualization tool will be discussed in contextually. And if there are any other resources, like sometimes you might not be able to have a good computational system, or there might be some difficulties, like if there is a protein with some metal, or or some ligand, or sometimes we need to understand interaction between protein and protein. So to these these type of interactions will be requiring different tools. I will be discussing all those things uh, relevant to at that context. And next is after these discussions and installations, I will be providing you some like questionnaire type, or practice or assignment, practice questions or assignments, so that you perform them. If you are not able to solve them, some of us I will ask someone to share their experience or how they are going to perform that. Otherwise I will be helping you to how helping solving those assignments, so that if you are doing it yourself, you will not forget, and you will understand it more. And one thing I want to make you all clear, that if if there is someone who is not able to understand things, I will be delaying the session, because I want to make each and every one of us to understand all these things. Next, if anyone has any question or doubt, can ask me right now. Hello.
Hello.
Yes.
Hello.
Yeah, you are audible. Am I not audible?
For how much time each session will be? Yeah,
Like,
yeah. You are too audible.
okay.
Yeah.
So,
For how much time? For how much time each session will be?
see, as I have already told, time minimum that will be around 30 minutes. And if some concept is going to be cleared in within like 10 minutes, 5 minutes, we will be having discussion session, so that if anyone is having any query, can ask there. I will be breaking down all the session in basic parts. Like I have already discussed, first I will work on installation, then visualization. So if we run out of time during installation, means we take 30 minutes to make you understand for installation, we will be putting next part for the next session, so that we learn uh, together. We learn each and everything very clearly, and uh, there is no problem to anyone or no issue to anyone. This is not a very professional course. Just I will be focusing on hands on. I think it is clear for you. Minimum 30 minutes, and if after that, after my session, if anyone have any doubt, can stay, and later other people can leave. And I will try to provide recordings or the PowerPoint presentations, so that if you want to replicate it later, or you cannot immediately perform those hands on, you can try it later, practice later, so that no one will be in loss. Any other question? Can't I clear the ink? Is there any other question from anyone, or any other doubt? Any doubt? Okay. If there
Hello.
is any
Excuse me,
doubt,
I have a question. Will we work with predicted structures? Like,
predicted structures.
if the structure is not known experimentally.
Okay.
Yes, sir, yes.
First, we will be starting with the structure which are already present, and sometimes we will need to modify structures, or we will need to find structure which are not already in domain. So I will be sharing the resources and walking you through how to get those structures. Like there are advancement in prediction using artificial intelligence, and there are multiple other resources or free web servers which perform homology modeling. Actually, uh, artificial intelligence based models do not perform homology modeling, so those structures are a bit different from actual homology modeling based structures. So I will be also taking sessions on those things as well. I will be trying to share all the resources I have collected or I have gone through contextually, from zero to like at each and every step you will be getting resources relevant to that thing. There is it, there it is. Why we will be taking case studies from papers. So your problem, like PDB structure for particular disease, that will be involving some pathway and network analysis type thing. I will be taking basics of those things, so that you can understand how you are going to get those structures which are involved in some pathway or network. So don't worry about that. Means you will be getting all kind of like how you are going to do drug discovery type courses. That complete package, complete course you will be getting from here. Any other question?
Okay, thank you. Thank you, sir.
You're welcome. If
Hello.
you want, you can call me Vishal. I am also a student.
Okay, Vishal. So I have a doubt.
Yeah.
How accurate is a molecular docking method when compared to the actual biological process?
Docking methods are like these are simulations. So out of thousand or out of
Okay.
millions, there might be one simulation study which could be near or near accurate to the actual in vitro experiment, but it helps us understand that those particular interactions are possible or not. If there will be any possibility like 1%, 2%, that will be ruled out in that we will be getting from molecular docking studies. So based on that, we will not be having any time wastage or resource wastage in vitro experiments. I am
Okay.
still having
Thanks.
one question, that why I am taking these sessions for free. Actually, just actually what happened, there are my batchmates or my colleagues and some juniors in this session. So I was not able to take all those people at one place. That's why I arranged this, and they are also there. I don't know their name because I shared the links anonymously on through Telegram, so that we don't have all like hesitations. So anyone in this session can ask any question or any kind of doubt. Don't think that I don't know this thing or that thing. Please be frank and ask anything you want related to docking dynamics or this biological processes or biology. So you are always free to ask me any question, and I would suggest whenever anything comes in your mind, like any doubt or question, you can put it in Telegram. I will be trying to answer all of your queries or try to find the resources or alternate of those things, those resources. And I think there are no more questions from you people. If there is any, please type in the Telegram group or here in the chat. Divyansu, I guess I have already answered your query about predicted structures. Is there any background noise from my side? There is some music session going on in my society, so
Yes, sir.
sorry for that.
Yes.
Do you have any question? Anyone? Can we end this session now?
Yes, sir.
Okay, thank you. So tomorrow we will be needing at least some desktop or some laptop or other system, so that you can install some basic softwares like Pymol, Chimera X, or other software. Basically, I will be walking you through some through theory and logics where you can apply it to any software like Discovery Studio or other visualization tools, or even some web based tools, so that you don't have any problem in understanding how any visualization tool is actually going to work. So this is kind of going to be session where you will not require any kind of help from others after this session. So you will be doing all those things yourself. Just be with me with your laptop or desktop. So I am ending this session. Thank you.
Hello,
Yeah. Do you have any question?
hello, sir. Actually, I want to ask that that the tools are have any charges for that
I will be mostly sharing.
that you are recommending the tools
I
like.
will be sharing free resources, and in Silco in Silco world. There, most of the software are free, but due to lack of knowledge, we are not able to use those free tools. So paid tools help us like drag and drop things. So we find those easy, but they are chargeable. I will be mostly sharing open source resources so that you can download them anywhere anytime and carry on your research. So mostly I will not be sharing any
Thank you, sir.
paid resource. I will not share any paid resource actually. I will tell you how things work so that you can use that knowledge in paid or free resource. Any other question, anyone, or any other doubt? Okay, I am ending the session. And if you have any query, please drop it in Telegram. And everything like uh, resources or questionnaire or time schedule will be shared through the Telegram group. Okay, have a good night, everyone. Bye.